All right, so I'm very proud to represent here in Poznan in an event co-organized by the Dioscuri Center to represent the TDA team from KTH. So, to describe what we've been involved in over the last several years, I thought I will tell you about my subjective, my personal view on TDA and its place. Okay. So imagine you came to this room and you see various numbers attached to objects here. No unit, just numbers. And your task is to figure out what these numbers mean. So the first thing you realize is that it's a major simplification of these complex objects you see. You assign a number to this chair. Okay. So what is it that you do with these simplifications? Okay. Maybe you observe that some of these numbers are very tiny, some of these numbers are very large, and you might think, maybe I should look at log scales of these numbers. Or maybe you notice some periodicity and you discover that maybe it's good to look at these numbers as elements of a circle or some other ways of representing this thing. This step I called modeling, because you want to transfer. Maybe the word modeling is not really appropriate, but you want to transfer these numbers into objects, possibly numbers, which are more amenable for statistical analysis. Once you do that, you do analysis. You group objects that seems to be similar, and then you look at the distributions of these numbers, or you gather objects that have similar numbers, and then look, what's common about this. You maybe know something, so you devise some learning schemes. You optimize some functions on them. You do analysis, whatever you have at your disposal, and you come up with some answer. And you maybe decide that it's the length of these objects. But see, I came before and I wrote the intensity of the color of these objects, and maybe in this room there is a strong correlation between the intensity of the color and the length. And although you are wrong for all practical purposes, you're right. Now you change the room to seemingly very same environment where it's different color scheme and you are wrong. Okay. So what I just described to you is an analysis pipeline, and for me, data analysis is study the space of this analysis pipeline. And data analysis, comparing to the entire mathematics, in terms of number of people, financial resources, interest, it's enormous. Math is really tiny subject comparing to data analysis. So in this vast universe of data analysis, there's this one little point, which is very, very tiny, which is called topological data analysis. And my aim for this talk is to somehow tell you why I think that sometimes it is worth going close to that little point, visiting that little point, this vast universe. Okay? So what is topological data analysis? Well, it's part of data analysis, so it's a subset of certain analysis pipeline. So what are these pipelines? What are these pipelines? So the first thing is that your input starts typically with a maybe broadly understood notion of a distance. Okay? Very broadly understood. Now the next step is that the simplifying representation is obtained using homology. Okay. All right. Now why homology? Maybe I will try to explain later. But the trouble with homology is that it's not really amenable for statistical analysis. Homology was designed to describe what I call control topology, for example, how cells, how Schubert varieties are built out of cells, how projective how projective spaces, cell decomposition of projective space. Homology has been very effective at doing this. Topologists have not looked at, what is expected homology. Homology turns continuous maps, simplicial maps into linear functions. And if I give you a bunch of linear functions and ask what is their average, I have no idea. Even if you turn them into matrices, which bases, maybe you are working over finite field. We do not know. We do not know what is the average. If I give you a bunch of simplicial complexes and I ask what's the average, what's the expected homology between these maps, between the simplicial complexes, I have no idea. So homology is definitely not designed for doing statistics. So I think one of the first key achievements, like major achievements in topological data analysis, that people found ways of converting that information into something that, into objects that are amenable for statistical analysis. And here, Peter's landscapes, persistent images. There are various ways of doing this, but ability of doing this and realizing that it is possible and meaningful, for me, it's one of those important steps. Okay? So, my aim for this talk is to somehow argue that it is worth spending some time, maybe in this little tiny area of data analysis, which is called topological data analysis. So let's try to go through first step. Why homology? What is so special about homology that it appears in this context? Okay. So let's discuss this. First step, you start with with distances, and the first step you do is to geometrize this. And one of my favorite, you convert that distances into time series of simplicial complexes. For example, one of my favorites is Vietoris-Rips construction. I will not tell you what it is, you probably know, but what I'm more interested in is why Vietoris-Rips is so special in this context. Now, remember, data analysis is about simplifying. I mean, we don't need, if we could understand the original world, like we don't need data analysis, we just stay in that world. The world is too complex. We need to simplify, and drastically. We need to assign number to this big thing, and maybe these numbers sometimes for various purposes can be useful. Simplification is the key. Now, that step is not a simplifying step. Geometrizing, for example, using Vietoris-Rips, doesn't add or subtract any information from a distance space. Worse, distance, maybe you can record it, as an array of distances. Vietoris-Rips complexes you cannot store on the computer. So this is like, unzipping. It's very spongy, contains the same amount of information, but it's like, it's unzipping. It's very spongy. It's not a simplifier. But it has one aspect that is very crucial here. So let's imagine that we have an experiment whose outcome, every time we perform an experiment, outcome is a Lipschitz function, okay, between distance space. So you perform that experiment twice. You don't expect to receive the same outcome, but you might wonder, how different are the outcomes of the same experiment. So you might try to estimate, how much they differ. Okay? So if you geometrize that information, you apply Vietoris-Rips construction or any other construction that you want. Maybe F and G, they are not the same, but if you move a little bit in time by that error, what you get homotopic maps. Vietoris-Rips construction has this property that it converts imprecision into homotopy relation. imprecision noise is converted into a relation that, it's right up to homotopy. Something that, topologists have been studied for a very long time. Okay? That's a key thing. Any other construction has that property. Noise, imprecision, is converted into a geometric relation, in this case, being homotopy. So now when you talk to homotopy theorists and ask, if I give you two maps, can you decide whether those two maps are homotopic? That's very hard. Okay, that's very hard. So we have to simplify. So we often hit this with homology, or we can hit this with any homotopy invariant construction, maybe K-theory. Okay, homology has these properties that two homotopic maps. If F and G are homotopics, these linear maps are becoming the same. Okay? So if you take this whole two-step process, we know that noise is converted into homotopy relation, is converted into equality, and we achieve denoising. In fact, that's how that's why I do often try to check. I do some experiment, I calculate some matrices. If they are not the same, the noise has to be larger. Those things you can do this. And I want to just also stress that this is a simplification. Although the time series of simplicial complexes is a very complex universe, the time series of vector spaces are much easier universe because of the barcoding. Okay, we achieve simplification. Now, what I also want to stress out that in this first step of this TDA pipeline, there's mathematical aspects of this, but there's equally important computational aspect. Whatever you come up with, whatever your favorite homotopy invariant construction is, maybe mathematically it could be interesting to study, but for purposes of applications, you have to come up with constructions that eventually you have to implement and use. And I think this is another point where TDA had a breakthrough, where people started, when the software was written, very effective, fast, well documented, where we could, and this is non-trivial. Like this part, if you are a mathematician, you think certain things are easy, you are very quickly confronted with a wall. That's another milestone that I believe made this thing useful. Okay, you cannot. All right, so this first step in this pipeline, what homology buys you certain control of noise. It buys you some, I would say, very exotic and unusual denoising properties. Something that it's very hard to come up using something else. Okay. So now the second step is you want to transform that information, this time series of vector spaces, into something that you can do statistics. And there are various options. You can do landscape, persistent images. There are various options. Today I will tell you about something what I call hierarchical stabilization. Okay. Now, when you apply the very first step, you start with a distance. You apply persistent homology. That's the first step. You can do two things. One, you can look at this individual time series and decompose it. Just look at this individual object. Or, which I think is more appropriate, let's look at this object as a part of that space. And we transfer this very complex situation into this more easier universe, let's try, it is not about this individual object, it's about how this object lives in this universe, how it compares to other objects, not individual. So hierarchical stabilization is about understanding the geometry, this entire geometry of this time series of vectors. Try to extract information about, not individual object, but how individual object embeds into that space. In a similar way to Yasu's talk yesterday, it was quite revealing for me because he wanted to understand the global properties of that universe that you try to translate some complex information into. This global, understanding that universe is very important. All right, so hierarchical stabilization. That's a known universe, that's a space, it's a time series of vector spaces. So hierarchical stabilization. All right, so you are somewhere in a known universe, okay? What is it that you might do first in order to familiarize yourself with this? Maybe you start looking at objects and it's totally dark. So you start taking an individual object and maybe look at number of handles it has. Okay. So you have your universe, a known universe, and maybe the first step is you get some functions to each object in your universe. You assign something simple, some number. Oh, this has five handles, or this has 7 components. Okay, some very simple operation. Typically, I want this to be easily calculated. Okay. All right. So, what is the next thing that you might do at that universe? Maybe after a while, you learn how to measure distances between these objects. Maybe one thing is here, the other thing is there. So, you choose a distance. And already these 2 pieces of information allows you to model that universe. To transform this unknown universe into a place that is much more familiar. So that information I will use that information to construct a map from this unknown universe T into much easier universe of real functions. And here's the definition. Maybe let me do this. What you do, you take an object in your unknown universe, and you want to convert it into a function. So you have to tell you what is the value at t. So you look at this object in your universe. You look at this distance at your space. You look at the ball, and you minimize your counting invariant on that ball. That's the value of that function. Okay. So it uses the geometry to define that function. I have to understand the geometry of the balls in that universe. Okay, so it tells me some information about how that point is embedded in that universe with respect to the distance. So what's amazing about this is that this function, this modeling, is Lipschitz. Here, this is chosen distance, and here it's more familiar, let's say LP distance. I often take L2 distance because it's a Hilbert space, so I can also measure this angles. These 2 pieces of information allows you to transform this unknown universe to look at the points, objects in the unknown universe as functions. Okay? Now, functions, we have a rich probability theory, statistics, analysis. We can do a lot of things with these functions. So instead of these unknown objects, I represent by functions. Moreover, we see that the key point, you have an important parameter of doing that, distances. Depending on the distance, I will represent your object in different way. Each distance will tell me what is the global geometry of that of this unknown universe. And according to that geometry, we have the shadows of these objects. Each distance gives you a window and some information, and that function will give you that some information about that, the geometry of how that point is embedded with respect to that chosen distance. All right. So our unknown universe is this time series of vector spaces. So for us, okay, all right. So if you have a time series of vector spaces, you know, vector space, you can choose a base. Time series of vector space, we have three operations, that not only addition of vectors and multiplying by a number, by your scalar, but also you can move in time. Okay? So what is a rank? It's the minimal number of elements, such that every other element can be obtained as, you know, using those three operations: addition, multiplication by scalar, and moving in time. Okay? This is called the rank, and it's one thing that you should know, that it's easy to calculate. If I give you a time series of vector spaces, it's easy to calculate that number. It's fast. Now, one of the key things in our activities was realization that this time series of vector spaces have a very rich space of distances. It's not just interleaving distance or Wasserstein distance. In fact, any function with positive values leads to a distance, and that's what was the subject of the workshop talk I gave. This workshop, 2 hours talk, so maybe you sort of remember. We talked about distances on persistent modules, on tame functors. Okay? This is very important. So I can, you know, the space of distances on this time series of vector spaces, you know, contains these very rich. It's very rich. This you know, every positive function leads to a distance. Okay. So you have for each such parameter, you choose a function. Here in this talk, I'm not telling you how to get the distance. Maybe those people who remember the workshop might remember. For each such function, you can model. Each such function gives you a window of representing your time series by functions. Okay, so hierarchical stabilization of the range. Each such function gives you a window, gives you a way of thinking about your time series as functions. Okay? And it uses, the most important thing, again, I will repeat it again, it uses the geometry of the space of time series, not individual element, that I try to decompose this individual element into some pieces. I look how this time series looks in that space of all time series of vectors. Okay. Now that representation is effective in the following way. Time series of vector spaces are isomorphic if and only if, for every positive function, for every distance, the shadows are the same. So if you want to distinguish your time series of vector spaces, if you have enough windows, you actually will distinguish. I want to emphasize that so far I have not mentioned, okay, well there was once barcoding. But what I find fascinating is that, you know, barcoding is about this individual guy. This says that I can recover barcoding from the geometry of this space, of these time series in that space. Barcoding is somehow about the geometry. Of course, barcoding is useful for algorithms, for, you know, effective calculations. But mathematically, you know, to me, what's really is important is that these invariants are really about the geometry of that space and how individual points are embedded into that space.
Now I can't remember the next slide. All right. So modeling is done by this by stabilizing the rank, and the key point is that we have a rich space of parameters. Now imagine that you have one experiment which is about, you have some biomedical data, and you have another experiment entirely different, some industrial data. Okay, some tests of industrial machines. Now, because they take normalization, they take other things, it could happen that this data, these numbers that you get, are very similar to each other. One coming from biology, one coming from industrial testing. So, if you did not have this rich space of parameters, if you just use interleaving distance or Wasserstein distance, you would treat both of these data in the same way. And maybe for biological experiments or biological data, this function should be different. Maybe the space in which you want to, the noise, the way these biological experiments fluctuates, you should design different F for that experiment and different F for your industrial. Why should we use the same measure for both data that comes from entirely different world? Okay. So the fact that you see now what happens is that I'm describing this little tiny patch of TDA in this huge vast universe of data analysis. But if you zoom in, you actually see some continuity there. You see all these the space of maps. Okay, so it's not really like a point. It's it's some continuum there. It's still very tiny, but it's continuum. Okay. All right. So this is called I call this homological modeling. Okay, homological modeling. All right. And now once you do this, we know that such functions, we have lots of, it's a Hilbert space. That was, I think, Peter Bubenig's observation that we have to move. The best scenario would be if we move that information to a Hilbert space. So with L2 distance, this is a Hilbert space. So you can measure angles. You have some data sets, some distances, and you can start by doing this. You have two distances, you can suddenly measure angles between two different inputs. Okay, you can do lots of things. So you do statistical. It's some vectorization of your data. Some vectorization of your data. All right. So in reality, the situation, the way we apply this thing is a little bit more complex. All right, because we have many other constructions, for example, Czech or some, witness versions or alpha complexes. We have some other constructions. Moreover, we might use homology or some other, we might be interested in some other invariants there, as long as there are homotopy invariant. We also have many other functions. We can look at this time series of vector spaces. We can take a snapshot. We can look at there are many functions. We could look at Betti curves, Euler characteristic curves. We can don't even go there. Maybe you are interested in counting invariants. People, many people count number of simplices that satisfy some properties. Okay, there are lots of other functions. Moreover, we are never interested in single distance space. Often what we have a point process of distance space. We have an experiment, we repeat this experiment, and we have a lot of them. Almost never happens individually. So the key thing, of this homotopy invariance, and that's what I want to reiterate again, why suddenly this topology, this algebraic topology becomes relevant in this data analysis. It's because, yeah, homotopy invariance, homotopy relation has been studied for a long time, and this homotopy relation is responsible for the fact that all these compositions, they may not be continuous, but they are always measurable. So you can take, you can take their expected values, you can take, everything that measurability buys you. But again, homotopy invariance, that thing gives you measurability, sometimes continuity, not every time, but measurability. Okay. So this homological modeling, we have these, in our TDA little patch, many of these points are actually measurable. All right, so let me try to illustrate on a very basic example of Henry Himeki. Let me just mention this. So, PhD in mathematics, done a lot of cool applications in neuroscience, for example, and many other applications. Currently in the Physics Institute and doing very intriguing mathematics. Maybe some of you had a chance of listening to Henry a couple of days ago here. Okay? So this is a little tidbit from his thesis. He considered 6-point processes on the unit square, each contain roughly 200 points. Yeah, I don't know whether you can see this. I just draw them. Some of these processes you can distinguish by eye that they are different. The underlying distributions are different. Some of them is less. So we took 5. We can produce them. We took 5. He produced 500 instances of each. Used 200 for training, 300 for testing, and we did this. This is to illustrate something. This is so what we did in this 200 for each of these point cloud. He produced a stable rank, and then he for those 200 training he took the average of these stable ranks, and he used that as a classifier. So then for those 300. He calculated stable rank, and he calculated the distance of this stable rank to one of those averages, and whoever was close was the level. Okay. So with the standard distance, so if we choose now we are choosing f, right? We are choosing the window in which we are going to sort of construct this stable rank. Okay. So with the standard distance, these are the averages for those 6 processes. And when you look at the and this is validated result. When you look at the classification rate, those are the numbers. Okay. Now let's change the distance. Let's change the window to this one. And after again validation, this is the improved classification. Very simple classification, and you can see that it's. You get a few percentage points better, and that's what we actually observe on many occasions. If you choose different windows, more appropriate windows, you improve, various tasks. Okay. Sometimes it's actually good to look at various windows, not just one. There's no one good window. Sometimes you look at various different perspectives, and you try to accumulate those perspectives. And sometimes this is more interesting. All right. So you see, once I just gave you an example of some points in that universe of data analysis. Once you have a point, you can start pushing yourself. You can start moving in that space. Okay. And one way to do this. So if you have your distance space, what could be a course of action for this distance space? Well, you could look at it globally and calculate its homology or persistent homology. Or maybe you might ask, why don't I look at 30 element subsets of your space. And you ask, you know, how do 30 element subsets in your space look like? On what's the expected, you know, geometry of 30 element subsets? Maybe that is more informative than looking at the global picture of your space. So you sample according to some rule, and now for each of the sample, you use your point that you have in your analysis, you know, data analysis landscape. You represent each of these subsample by a function, and then you take average. And at the end, you represent your distance not globally, but you represent like what's the average, you know, expected geometry or shadow of, I know, 30 element subset. Okay. So you increase. This is another homological modeling. So in your space, you're like pushing it outside. You do it, you know, it's still very tiny. You push it slightly. It's still tiny, but you push it a little bit. Okay. So let me illustrate what it does. When I talk to some life scientists, I like to tell them that we try to look for geometrical invariance. Now, how do we, like, what does it really mean? You know, if you have a data which is 20,000 dimensional, what does it mean geometry? The reason I tell them it's geometry because I apply this to some very simple situations in which, you know, I visually can say, oh, it's something geometric. And then, because, you know, it happens in 2D or 3D, I say, oh, it's probably, you know, that's why I use geometry, you know, in 20,000 dimensional space as well. Okay. And here is what I do, and I show you this averages, and the ability of taking averages is important. So, you know, this is a figure with 1,000 points, you know, with some random normal error. Among those, I choose 100 points. Some, you know, I don't know how many times, 40 times. Then I calculate. This is a standard noise. I calculate H0 and H1. Those are functions. So I can divide that one by the other one. Okay. This is quite common, like normalization. Okay. I take average and I record on that side. Okay. I record on that side. And here is how it changes. You can sort of see, first of all, you can see that it is not about homotopy theory. It picks up certain geometry. Because, you know, on one hand side, most of those figures have the same homotopy type. But, you know, so it picks up geometrical thing, you see continuity, and, you know, you get an argument that picks up some geometry. Okay? All right, so let's look at a toy example. I give you another toy example. I have you know, 200 images, some point clouds. Some of them are labeled with blue, some of them are labeled with red. Okay. It's and you have to figure out, you know, how they split. When I draw them, like all of them together, you can sort of see. You can sort of see that, you know, what's the difference. The way I generate them is that up to 30% of points, and that can vary between 0 and 30, up to 30% of points is noise. I generate some points according to randomly chosen distribution. So I don't even know what distribution is, it's up to 30%. And the rest of it is taken from either a circle or, you know, this foreground, which I really try to fit that foreground to the circle. You know, I really try to fit that. Okay. I want to make a point. You see, I want to make a certain point here. So remember, so this whole plan is to model your data, this complex data. Although this is two-dimensional, it's not actually complex, but complex data by functions. Okay. So here is the image. Instead of these pictures, here's the image of these functions, blue and red. Now, and here's a warning, the space of functions is sort of weird. Because if I would draw that thing, I might, by visual expectations of these functions, I might decide, well, I don't know whether this, looking at this picture, is like distinguishing blue from red, for example. But the space of function is weird. Oh, okay. I do it for H1, sorry, I do it for H1. And when I do it for H1, actually this, for me, visually, it's better. I can somehow see that, that red gets separated from blue much better. Okay? Now, this space is a Hilbert space. I can measure angles between these functions. So I can support vector machines. This is a linear classifier, but why not? And I get 82% classification. Okay. Now, if you try to look at angles between these functions, functions are very weird. You might miss them because they sometimes go faster, some of them come later. Actually, SVM, angles between these functions, SVM is much better. So we have actually lots of examples in which, when I visually, I cannot, see anything. But so what I want to say is that, functions maybe are not, at least for our visual expectations, is not maybe the best universe. But if you are equipped with some tools, you might be quite effective. Also, I want to that if you choose the right distance, the right window of representing this data, you actually get up to like 95% accuracy. Okay. 95% accuracy. Okay. Here's another example of the things we've done. Insight into microglia morphologies. Jens Agerberg, who gave a poster and 10-minute presentation, talked much more in details about this data set. So we are collaborating with Sandra Siegert's group at IST in Austria, and she collects a lot of morphologies of microglia cells. Okay? These microglia cells are very heterogeneous because they depend on the environmental cues. They don't really know what microglia do. They know that they have some immune purposes. They, for example, if you're a mitochondria that produces energy, and the cost of that is producing some impurities, microglia would absorb these things. They are very active. Some, when there's some degeneration or some action, maybe some physical damage, they would be very active. They swell, they go. So when they kill a mouse, they take a slice, take a picture. These microglia would be in different state of their activity. And they had questions. For example, do microglia between males and females differ? Do microglia between different parts of the brain differ? Do microglia, and again, because they are very heterogeneous, it's not like individual, right? You have to do some averaging. On average, expected microglia in male are different than expected microglia in females. Now, I do not know why these questions are interesting. Okay, like if someone asks me, I do not know. People maybe wants to learn about microglia, anything, because there's some maybe suspicion of relation to some disease, etc. So we applied homological modeling, not to actually morphologies that these morphologies were transformed by this TMT that Pavel was involved in. And we could answer their questions. We look at these, we represent them by certain average stable ranks. This is those are functions. So we can, some vectorization. So let's just look at this UMAP, like two-dimensional dimensionality reduction, and it definitely shows the difference in where they come from. Okay. This is just an illustration, okay, of what it can be used. And again, ability of doing statistics has been crucial. Okay, has been crucial. Okay, let's look at another data set. When you go to life science conferences, you know, very often these conferences about the newest technology. And right now, 70% or more talks are about single cell transcriptomics and high C technology. Well, two things that would be 70%. So what is high C? So you look at DNA strand. If you actually make it flat, it's almost 2 meters long. Now each gene, each gene comes with a promoter. It's a little piece of DNA strand before each gene, and this promoter, you know, acts, you know, to tell the system whether that gene should be expressed or not. Now each promoter interacts with other parts of the genome called enhancers. The enhancers are non-coding, so they do not express, they do not code any protein. Are not related to a gene. But what happens is that these enhancers, each promoter acts with several enhancers, and these enhancers are related to a single promoter. Okay, it goes that way. These promoters are like switches, and they can tell whether that gene should be expressed or not by affecting that promoter, by interacting with that promoter. Now, physically, what it means that that strand in your chromosome, when it's embedded, that piece of the genome enhancer is physically close to the promoter. So that information, the technologies that they color it, but they freeze a cell, they would freeze a cell, and they would cut it, and they would count the number of these frozen connections. So enhancer interacts with promoter if it's physically close. Now what I did not know, for example, is that the way your DNA strand is embedded in your neural cells is different than they are embedded, how they are embedded in your liver, for example. Because that embedding will tell you that these cells actually, function, in different way. They produce different proteins, etc. So typically typically the experiment would take around 2 million cells from particular tissue, 2 million cells. They would do this thing and they would say, oh, we found 21 interactions. That promoter interacted with that enhancer 21 times. And then I'm like freaking out because I say 2 million cells, if 21 of my 2 million cells die, I will stop producing some protein. Okay? But they say that actually, it's all about statistics. In fact, the DNA, it's not rigid, it actually fluctuates. So when they actually take this thing, some of them go up and down, they would repeat several experiments, and they statistically would say, even this 21 among 2 million, which I find a little scary, it says that statistically significant, and I should assume that there is that interaction. I am a mathematician, I won't question that. My data was this information. So what we did, okay, by the way, we had 6 cells. It's very costly to produce them. They have maybe 50 more cells, but it's very costly, expensive, and time consuming. We had 6 cells, 2 repetition of various developing stages, from pluripotent to adult, 3 stages of development. And the question to us was, can we see the differences in the stages of development? Do pluripotent cell that is the cell that can probably become any other cell, does that cell, is that interacting between promoters and enhancer different from the development. And of course, you would like to sort of argue that these two cells will have the same geometry, these two cells will have the same geometry, but they are different. So what we did, we look at that information and we turn it into a distance, sort of graph distance. You can go from promoter, you look at the distance by actual physical distance to the enhancer, or you can jump to the enhancer. Okay? So we look at that distance. And here, maybe I'll show you this. So here is modeling using stable rank of these 6 cells. And you can see, for example, here the per chromosome, in chromosome 7, that blue cell is actually very close to the adult cell. Okay? But if you go to, let's say, chromosome 1, it's a really nice separation. Chromosome 2, 3, again, it's a little bit confusing. 4, it's again, rather nice separation. If you look, for example, here, well, here's not such a nice separation. Here's quite a lot. This is chromosome 10. Definitely, you can definitely see a separation between them. Now, in this case, and there was a basic question to us, but they are not really, they are also interested in this, but they are more interested into understanding whether particular promoters or particular gene changes, is the role of that particular gene changes with the stages of development. And they have, so they're not interested in maybe this global thing, but they want to understand how particular promoter or gene changes with the development. Not the global, but particular. So here we can explore the geometry of this space of promoters and enhancers. So that's your data. So here's another one. I just showed you, if we have a data analysis pipeline, topological data analysis pipeline, I showed you how we can sort of expand it. I will expand it even further. This is your data. So you took some points in your data, and you might wonder, how does that point, this one big black dot, embeds in your geometry? We do the same thing. So you look at the neighborhood, let's say the neighborhood, and you ask, how is the neighborhood of that point looks like? So I can apply your homological modeling. For each point of your data, you look at its neighborhood, and you represent the geometry of that neighborhood with some of your favorite homological modeling. So you get a function. But you see, maybe your space is very homogeneous locally, and what distinguishes between points, it's not what lies next to you, but it's the valleys around them. So maybe instead of these neighborhood, you might take this neighborhood, this blue neighborhood. Maybe that's what distinguishes. Locally, they look sort of the same, but the valleys built around them. So again, you can model these neighborhoods. And then your data set, you take your data set, again, into a function. Somewhat different. It shows you how the points embed in your space by looking at the values. Well, you can combine those things, and you can model by 2 functions. Okay? By 2 functions. So you can, maybe you can choose 3 neighborhoods. So maybe you find 3 neighborhoods, and you match each point of the data. You model it by 3 functions. And then if you have 3 functions, you might ask, well, let me project this on R2. 3 functions are complex, so let me just find some projection. That's, for example, the distance of the first to the second and the first to the third. And then you map this a little more, 3 functions into, your dimensionality reduction. Okay? So in that case, what we did, we look at 22 different regions for each of the promoter. Now I do it individually for each of the promoter. We look at 22 different geometrical regions of that space. We took H0 and H1. Okay? So, yeah, so here, chromosome 10, promoter 46 in all the cells. And you can see that even the geometry of that, and I took I go into 44, but I project this into F3, F5, and 10, and you can see that actually you can see a difference between even the role of that promoter. Now what's interesting is that they actually have expression levels. So it will be very interesting to see whether the differences here are correlated to the way these genes are expressed in these animals. So let's look at this reduction, this dimensionality reduction, and you can see that the space of promoters fits, has some geometry. Now, I've done a lot of these pictures of this modeling, and you very rarely get pictures that show some geometry. It will be like a blob, or maybe things lying on the diagonal because these functions are strongly correlated. It's very rarely, it happens once in a while, so that means that it does. So this was done for one cell. Let's just do repetition. This was done for the pluripotent cell. Let's look at the other pluripotent cell. And it's, this pattern is very close. Now let's look at the next age, next, and you can see that they actually would be different. Okay, these projections would be different. So now we are working, the next step is to combine this information with actually expressions and whether maybe protein productions, etc. Also, we will have some access to degenerations, to certain diseases, and which they know are related to the wrong embedding of the genome. And the question is whether we will be able to, whether the geometry, can see that differences. Okay. So let me finish with the last example. Pfizer released, it's now approved, a new treatment for this horrible kidney cancer. It's very deadly. There was a previous treatment, and they released a new one. The trial was, phase 3 was on the 726 patients. And that, you see, at the phase 3, the primary, the reason to approve that medication is to show survival rate. Survival rate is typically illustrated by something called Kaplan-Meier graphs. And you want to, the blue one is the new therapy. You want to make sure that that blue is as far from the red. Okay. Now, the reason this treatment was approved was because of that picture. That's the entire reason. They did also, they've done this information, and that's the outcome after this testing. Now, what we've done, we look at this data. They release all this data because the drug was approved. We look at the expression geometry of the patients. So you took, so expression information, no clinical information. We just only explored the expression data. And we wonder, can we use this expression to identify patient responders? Those patients that respond better to this therapy. So if you can do that, then you take a biopsy from a new patient and you see whether that new expression fits with your prediction or not. Okay? So we use this homological modeling. We transformed this data into functions, and we explore the geometry of these functions, and we notice that these functions, there are three groups. There are three groups of patients, entirely determined by how the geometry of that space of this expression vectors are. Okay? And then we checked clinical information. What is so special about these patients? And it turns out that in all these groups, the survival rate, the Kaplan-Meier, were much better than for the entire cohort. So definitely these people should be prescribed this new therapy. But because the Kaplan-Meier was so improved for these three groups, for non-responders, which is another half, actually the Kaplan-Meier was not. It was similar or even worse. So these patients definitely should be prescribed this new therapy. The rest of it, it's unclear. Plus, you know, using you have possible yeah. You can use it maybe to possibly predict whether a patient is responding or responsive. I think this is it. Thank you so much for your attention.